Hello! Today we are going over the axolotl. Hope you guys enjoyed the video of my little baby feeding little wormies. But yeah, we're gonna go over these guys. They're pretty cool. If you haven't done this already, do so. Big points from this. These axolotls are pretty cool because they're what's called neotenic. Neotenic. What that means is instead of metamorphosizing like most amphibians, they actually retain juvenile features, which is why they have little frilly gills. And then they remain aquatic. So they're pretty cute, but they still have those little legs that like enable them to stand up on their surface, but they still remain aquatic. They even have fins and everything. They get legs, but other than that, that's it. They're like fish with legs. They're super cute also. But what's really cool about them in terms of cancer and stem cells is that they have regenerative properties, meaning that over and over again, they will perfectly regenerate their limbs and body parts. And like I was talking about in the video, you can take a cell from one axolotl and a stem cell and put it into another body part on another organism, and it will completely regenerate where that stem cell was from. And they're what's called pluripotent. That was basically a big word that just means it can only become a certain type of cells. So if you were to take an axolotl arm, like if you cut off my arm, I was an axolotl, you cut off my arm, and you were to take I would be able to grow back this arm. Whoop, pretty cool. And if you took one of my stem cells from that arm injury and put it into another axolotl, it would also be able to grow an arm. Okay, it wouldn't be able to become a head, but it would be able to become an arm. So they're specific to the limbs that you take them from. It even talked about, which is kind of weird, they took a stem cell from a head of an axolotl, and they could actually put it into another body of an axolotl, and they could grow a head out from the side, which is really bizarre. But what's unique about these guys is that our stem cells, once they are embryonic, once they become embryonic, they can become like when you're a tiny, tiny baby, fertilization just happened, not even a baby, a fetus. You're in your mom's uterus, okay? You can become any type of cell at a certain point. Okay, so we call that embryonic stem cells. They can become any type of cell. They're not specialized yet. They can become anything. But once it's determined, it can, once it's become that specialized cell, it can't go back. So once you have your your cells that become the skin on your fingers, it's determined to become that. It can't go back to being a stem cell. It is going to be a skin cell for life. But this is where axolotls are a little bit different. This is why scientists look into these guys. One reason is because they can actually retain their stem cell ability. So you can take it, that's why you can take an arm cell from an axolotl, put it onto another one, and it will regrow that arm. Super weird. But that's why they're interesting. So they can become any tissue. They can become any of that type of tissue in that arm. So it can regrow bone, it can regrow skin, it can regrow muscle, but it can make an entire arm, an entire limb. It can become anything to make this arm. It cannot make like a leg. It cannot make a head. It is specific to that body type. But they retain their stem cell ability, which is weird. What could we learn from these guys is that you can regenerate, maybe from cancer patient perspective, we can learn how their regeneration happens cellularly, and then we can help cancer patients regenerate their normal cells. So once they go through chemotherapy and they're wiping out all of the cancer cells, they're also harming some of their normal cells. So you can actually go in and maybe regenerate more of those normal cells to retain functionality of each of those organs. That's one thing we could learn from this research. And if you remember that one part where they became glow in the dark, and basically you could just make certain types of, you could tag their cancer cells to make them glow in the dark because and only the cancer cells are glowing in the dark. You can do the same thing, and this is why we had them in the lab. You can do the same thing with virus-infected cells. You can take a cell, tag it to glow in the dark, tag a virus to glow in the dark, and you can actually see the virus spreading through the tissues of the axolotl because they're so see-through. So we used to look at several different see-through amphibian species to kind of watch how the virus spread. And then you could even see the virus being shed from those organisms as well. So meaning if I was an axolotl and I was infected with something and I was glowing in the dark, if I were to cough, you would see the glow in the dark viruses coming out of me because you're seeing the glow in the dark virus. So you can actually see how they spread, which is kind of cool. Okay, so that's a big takeaway from the axolotl. What you're going to do today is you have your Ebola disease detectives, okay? You should have this, and you should have a sheet with sequences on it, which today you're going to do part 1, so you will need those sequences. But all I did was, bear with me, there they are. I basically took that page with my DNA sequences. You can see the DNA letters. It has a reference sequence and then sequence 1 through 15. This reference sequence, and you're going to watch the video and do all of the first page and part 1. But just to recap what this reference sequence means, it is the original Ebola sequence. So if you see reference and you're wondering what that means, this is the original sequence from Guinea. And when you watch that video, the Ebola disease detectives video, this will make more sense. But the reference sequence is the Guinea original sequence. And then you're going to cut all these out, as I have here, cut each of them individually out and group them how you want to. And we're going to go over the best way to group them. But for now, you're just kind of investigating today. So today you're just cutting them out and you are going to group them up in groups where you think they belong. Okay, so what do you think the best way to group them up is? That's what you're figuring out. So looking at this, first, just to reiterate, cut those things out off your sheet of paper. You were given that, but I will also upload digital copies of all these things in case you lost it. But you have this. You're going to watch the video. It tells you to watch a video for background information. I'm going to upload that video to this post. You're going to answer these questions from the video. Then you're going to do this procedure, which is what I was just starting to go over. You're going to cut those out and group them into patterns. And you're going to do all of part 1, which is just this page. All of part 1 and the analysis questions. All these questions in part 1, do not do part 2. Don't do part 2. We're going to do that together tomorrow. All right. Good luck. Any questions? Send them my way.